Just now we have talked about the water movement in this animal cells, okay, which is in this case represented by our red blood cells. Okay, next we are going to look at the plant cells. So now it's actually the same steps that you will be using. Now the plant cells, they are placed in a hypotonic solution. Hypertonic solution. Now compare in the cells and outside the cells, the intracellular and the extracellular fluid. Compare their concentration. So if now the plant cells is now in a hypotonic solution, so that means this surrounding solution has got higher water potential as compared to the plant cells within the plant cells. So osmosis tells us that there will be net movement of water from region of high water potential. In this case, water will diffuse into the vacuoles. Still remember the vacuoles, and it is through osmosis. Okay, observation is that this is very important when you describe the plant cells. You need to include all these points over here, but it's not tough actually. If you understand the concept, you can go about it from the beginning. So having identified the concentration between these two regions, okay, having determined the movement or the direction of the water molecules by osmosis, now the third step is to look at the observation or to predict the observation. So now imagine water already diffused into the vacuole of the cells. So definitely the vacuoles will get bigger. So we use the word expand. Okay, if this gets bigger, the vacuole gets bigger, it's going to create pressure to the surroundings. So that means it's going to push the cytoplasm and the plasma membrane very near to the wall, against the cell wall. So imagine all these forces coming from within, pushing the cytoplasm, pushing the plasma membrane to the cell wall. Okay, now you need to put this term in where you say in this condition, the cells are in a condition of being turgid, segah, turgid. Okay, maksudnya dia tidak lembek, tidak flaccid. So we say the cells are now in turgid condition. But unlike this one, actually they are at the same condition right now. But unlike the red blood cells, these plant cells will not burst because their cell wall is rigid, tough. The cell wall is strong. They are made up of cellulose. Okay, the polysaccharide, which is real tough and strong, and that's the one that gives your plant cells the fixed shaped. So that's why your plant cells will not burst like the one in the red blood cells. Okay, they are now turgid. Imagine now they are standing strong, especially in herbaceous plants that do not have woody stems. Okay, imagine that. So we say turgor pressure is the one. Now, turgor pressure is important to plant cells. Okay, like what I've mentioned just now, it gives a support. It maintains the shape of the cell. And cell turgidity causes the gut cell to swell. The gut cell, remember the gut cell? The one which controls the opening and closing of the stomata. So when the gut cells is turgid, the stomata will open. And when the stomata opens, it is important for photosynthesis because this tiny pore is going to allow gaseous exchange to happen. Okay, and we know in photosynthesis carbon dioxide is needed, and therefore the opening of stomata enables these gases to diffuse into the leaves and hence making or increasing the rate of photosynthesis. Next is on the effect of the hypertonic solution. So now imagine just now was hypo, now in a hypertonic surrounding. So first step to do, identify the concentration of the two compartments And now that the cells are placed in a hypertonic solution, so that means the concentration, the intracellular fluid in the plant cells is now higher. Okay, the concentration, that means inside the cells, the plant cells has got higher water potential as compared to the surrounding hypertonic solution. Okay, hypertonic solution. So the net movement of water, it will be from a place of high water potential to lower water potential, isn't it? So this will cause the water to diffuse out of the vacuole. So now it's leaving the cells, going to the surrounding hypertonic solution in it. So the water will diffuse out of the vacuoles. Okay, don't forget the word by osmosis. Okay, so having done the first step, which is to identify the concentration of the solution, having determined the direction of the water movement, next is the observation. So here you need to tell or to predict the observation. So what happened to the vacuoles in the cytoplasm? So it's actually opposite the first one. So in this case, opposite will be vacuoles and the cytoplasm will shrink. Use the word will shrink. Okay, put in the terms vacuoles and cytoplasms. So then what happened to the plasma membrane? Compare. So in this case, plasma membrane will pull away, okay, pull away from the cell wall, pull away. So it's like further away from the cell wall, it's going to retract, but not fully. Okay, and then put in the terms plasmolysis. The plasmolysis, it's the name of a process. So what happened? What is plasmolysis? This is the one. If people ask you what is plasmolysis, okay, this is the process where, okay, then you describe this is the condition of this plasmolysed cell. Okay, the second statement is the condition of a plasmolysed cell. So plasmolysis, what is the effect? It's going to cause the leaf and the stem to bend downwards and wilt. Okay, so that means no longer turgid like the first one. Okay, it's going to wilt. So a plasmolysed plant cell can regain their turgidity. Okay, that means this effect can be reversed. How? Quickly treat the cells with hypertonic solution. Quickly treat. So that's why immediate. So you want the plasmolysed cell, you don't want this to happen, you want them to gain their turgidity. What you can do is that you replace the solution. Okay, now replace the solution with hypertonic solution. So what will happen is that water will diffuse into the cells again. And we say the cells are now undergoing, opposite this one, deplasmolysis. Deplasmolysis. Okay, is that clear? Okay, the last one to describe is the effect of isotonic solution. So when the sept, okay, hopefully you still remember what is the sept, the cell sept, which is in the vacuole. When the sept of the plant cell, so that means it's referring to the inner fluid, intracellular fluid, the one which is in the vacuole, and the extracellular solution are isotonic. That means in and out of the solution, the concentration, they are the same, they are isotonic towards one another. We say the water potential is the same. So what happened is that the movement of water diffusion in and out of the cells is the same. So in this case also we use the term flaccid. Okay, you use the term flaccid, not turgid, but not to the point of plasmolysed. Not to the point of plasmolysed. Okay, we say flaccid. Right? We don't use the term maintain their normal shape like the one in the animal cells. For plant cells, if they are in a isotonic solution, okay, the second statement very important. It says you must tell them this to show that you understand water diffusion in and out. They are the same. However, they are flaccid lah. They are flaccid. This is actually their normal condition. But they are not wilting, they are not plasmolized. This is just the correct term to use. Okay, and that you must tell that you understand that the water potential is the same. Okay, there are also experiments on the to study this one 3.3, 4, and 5. Okay, on these different types of solution. Okay.